All right, welcome biology B students. We're going to take a look at moving on into Mendelian genetics. We just finished up talking about meiosis and how those chromosomes are going to be separating out. Now we're actually going to take a little bit closer look and see what's actually on those chromosomes, in particular certain genes or traits that are going to be expressed based upon what is inherited within the gametes from your mom or your dad. Now, one of the individuals that started learning and thinking about this was a guy by the name of Gregor Mendel. He was an Austrian monk, and like many monks, had a lot of time on his hands. He'd studied certain pea plants in the gardens of his monastery for many years. You can see his birth date, 1822 to 1884. Gentleman that died kind of at an early age, but 60 some years old at that age. Maybe it's not too early. But he had published a theory of inheritance and talking about how things are inherited in 1865. But at that time, there wasn't a lot of people that actually agreed with a whole lot of the things that he stated. But after a while, in 1902, it's almost 40 years gone by, they realized, Mendel really did know what he was talking about. So later on, people now what they call rediscovered. They took a look more at his materials and realized that the stuff that he had in his published book was really true. Now, in his experiments, what he took a look at were certain experiments between cross-pollinating pea plants. And his focus worked on 7 different types of traits. Seed color, if the seed was actually yellow or green. Seed shape, smooth or wrinkled. Pod color, pod shape, green, yellow, or inflated or pinched. If it was tall or short, if the flower is actually at the very end, or if it's at the beginning. Flower seed coat color, if they're purple flowers or not, gray flowers, white seed coat, all sorts of different variations. What he did in his experiments was that he used mathematical ratios and he identified each phenotype. Now that term you're going to see here in a little bit really refers to the physical appearance. What does it look like? That's what it comes down to. Is it yellow or green? Is it green, yellow? Is it inflated, pinched? Is it tall, short, and so on and so forth? That's what a phenotype is. Okay? Move our way on to the next seed. So this is a whole bunch of different phenotypes that he was looking at. If you look at number 1, seed coat versus flower coat. 2, seed color. 3, seed shape, smooth or wrinkled seeds. Pod color, if it's yellow or green, inflated or pinched. Certain locations, if it's tall or short, and or where the flowers are at, if it's at the very beginning or if it's at the end of the actual flower plant. Now, what he started to take a look at were certain structures or components. Now, let's get off of this quick and bring this over just a little bit, and we'll shrink that. And take a look at the actual, see the whole thing in comparison. Now, some of the basic terminology we're going to look at are in terms of generations as well as crosses that we're going to do. Now, P generations, the first thing we're going to talk about is your actual parents. What do your parents have? Your parents have certain traits. What traits did they give you? And those traits that they give you are in you or yourself are considered their filial, their first generation, their offspring or progeny. So in terms of your parents, you got your mom and your dad. Right? Let's see, I don't know how to properly make this. You get your mom and dad, they get the progeny. They get you. You are considered their F1 generation. Now, you're probably way too young to think about this, and you never want to think about this right now, but when you decide to have children of your own, your children would be the offspring of your progeny. So they would be considered the F2 generation, or the second filial generation. The F stands for filial. So in terms of what we're going to see here later on, just kind of shortly squeeze this up and we can kind of come back to it. But in terms of an offspring, you're going to have the mom and the dad. Those guys are going to make you. And what this is I'm drawing right now is a pedigree. Circles represent female. Squares represent male. So there is the P generation. These guys are the F1. Now, say you are the female. You get married to some guy, and your kids are here. Again, you get one of each, boy and a girl. This offspring is considered the F2. Okay, so parental generation, F1, and F2 generation. That's what Mendel was starting to take a look at and trying to figure out what he could refer from it. Okay. The other things that he started to take a look at were in terms of, let's kind of get rid of that a little bit, was in terms of the crosses. He looked at crossing certain traits. If it was a single trait, we used the prefix mono. If they're only looking for one single thing, and this can be anything from widow's peak, if you have a hitchhiker's sum, a lot of times we use it in terms of like eye color, hair color, but those aren't really that simple. But there's a whole bunch of things, but if you're just looking at just one particular phenotype, in a dihybrid cross, we're actually trying to do two different phenotypes, so di referring to two. All right. In terms of genetics etiquette, just like most things, a little bit of chivalry. Female is usually written first. Move our way through. All right. Now, if I'm looking at this, we have what's called our Punnett square going to be developed, and we're going to talk about a dominant and recessive trait. Dominance traits are always going to be represented by a capital letter. A recessive trait is always going to be represented by a lowercase letter. So in this illustration, you're seeing capital S's and lowercase s's. There's a few other things we're talking about. This term here, alleles. Allele is the particular trait that you're referring to. So we have a capital S allele coming from that chromosome. That chromosome would have been the one that you got from either your mom or your dad. So if this is you, you might have gotten this one from mom, you might have gotten this one from dad. What they both had capital S's. They both had the same trait. And being that they're both capital, both the same, they are homozygous. But if this one over here was you, and you got two little ones, they're both the same still. But being that they are recessive, or little, they are going to be recessive trait. Now, in a complete dominant trait, one is completely dominant over the other one. Meaning if I had a big S from, say, mom, and a little s from dad, if this was you in the middle, luckily enough for me, I've got one of two options. This big S is going to be dominant over the little one. Now, one way I think about it, I think of like you playing basketball against a preschooler or a kindergartner, right? You should win every single time. No matter what the outcome should be, you always should win because you are the dominant trait. You are bigger, you're stronger. That's what's showing up here at the big S. What it's really going to do, it's going to mask the other one. The other one's going to be there, but it's going to mask. So what you're going to show is in terms of the actual physical characteristics of the dominant trait. So in this case, if big S represented smooth and little s is wrinkled, every time I see the big S, there should be a smooth seed. Now notice here, both of them are big S, two big S's, so it's got to be smooth. This one also has at least one big S, so I am also still smooth. However, this guy over here, there's no big S to be found. And the only way I can show up with a big S, the only way I can show up with this wrinkled phenotype is if they are both recessive, both little, and sure enough, that's the case. All right, so take a look at what Mendel did. He was looking at that P generation, those parent generation, and identifying that P generation, if I did a cross pollination with a smooth seed and a wrinkled seed. So in our last one, we know that this could either be two capital S's or one capital S and lowercase s. Not quite sure yet, but we know this one has to be this and this, both lowercase s's. What I'm going to do is I'm going to cross those, and that's what Mendel did. He said, well, let's cross these, one of those two, with one of this guy. What he found out is every single one all developed smooth seeds. Why? Why were they all developing smooth seeds? So in terms of a cross, what he ended up seeing is that if this was the case, if you were homozygous dominant, having two capital letters, and you crossed it with a wrinkle seed, every single time you did so, you're going to end up with smooth seeds, and that's shown over here. In a quick illustration, what that really refers to, and you're going to utilize this math tool, this little Punnett square. All this refers to is making a square and looking at the predictability of something going to be happening. And what that really refers to here is, so I'm just going to move that. If we're looking at the smooth seed, we said we could be two big S's, but you cross it with a wrinkled two little S's, you're going to put all those big S's down, and you always start with a capital first. So we got a big S, big S, big S, big S, and then you cross it. We're going to have little, 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 little. So 100% of the time, you're going to get this letter combination. That letter combination is called a genotype. The genotype is a letter combination. Now, if I'm looking at what do they all look like? If they have at least one big S, what should they be? Let's go back to our picture. If I'm looking at our picture here, no, notice that back of our picture that big S is referring to the smooth seeds. So all of them, if the genotype, should be smooth. And that smooth is represented as its phenome. What does it physically look like? Okay, so genotypes for big S, one little s, and 4 out of 4 are smooth. But notice my combinations. Are they both the same? Think to yourself, no. There's going to be one big, one little. And so what we're going to see here in just a second is that with that combination, if I cross those again, let's cross the offspring of that F1 generation. Now again, F1 is the kid generation. And what I end up with the kid generation were all of them like that, big, little. Now, that term, I think we talked about it a little bit before, is heterozygous. If they're too big, homozygous, or too little is homozygous. Now, our first Punnett square, we were crossing those two, and we got these two. Now, if I cross two that were like that, in my Punnett square, again, I drop my S's, I drop them from the top, and I drop these over. Remember that if there's a capital letter, that's always represented first. So in terms of my offspring, how many capital S's do I have? I've got, this box has got a capital S. So that one's got a capital S. This one's got a capital S. That one's got a capital S. So 3 out of the 4 are going to be what's considered smooth, because that is the dominant trait. This guy is not. So 1 out of 4 is wrinkled. Okay. So if I look over here at my actual phenotype, that's what should it say. 3 out of the 4 are actually smooth. 1 out of the 4 is wrinkled. And taking a look at the last part, that last part is illustrating crayon. So again, that last part is referring to that genotypes, looking at those genotypes are now showing you that you have got one of them are one big S, two of them are big S, and one little. So what you have is considered a 1 to 2 to 1 ratio of the genotype. And that's showing you there. Okay. All right, now this one of the last slides we're going to take a look at here quickly is just showing you, confirming the principle of segregation, how they do separate out. When we did divide these, every time we are looking at two homozygous working together, you'd always get 100% homozygous dominant. Two homozygous recessive, you'd always cross them, get 100% of those. But how would you know which trait is going to be dominant or not? You're going to always try to cross it with something that is the recessive trait. So if we were to cross a homozygous dominant with homozygous recessive, we should always show a dominant. If we cross it with heterozygous with homozygous recessive, we should always get half and half. Okay? Here's just showing you a little bit of a test cross, and we're going to actually complete